Welcome to StudentPal. In this Grade 12 Biotechnology lesson, we begin Chapter 8, Animal Cell Culture. Animal cells can be maintained outside the body when their nutrients, physical environment, attachment or suspension conditions, and contamination control are carefully managed. These cultures support vaccine and antibody production, virology, toxicity testing, disease models, and biomanufacturing. But a flask of cells is not a miniature whole organism. Cell origin, passage history, identity, viability, contamination status, and the limits of the model determine what an experiment can mean. By the end, you will be able to compare primary cultures and cell lines, adherent and suspension growth, different viability measurements and preservation methods, while explaining the quality, biosafety, and ethical controls required for responsible use. Our roadmap starts with balanced media containing salts, amino acids, vitamins, energy substrates, buffers, and growth-supporting factors. We then connect temperature, pH, carbon dioxide, oxygen, humidity, osmolality, and surface properties to cellular physiology. We examine the biosafety cabinet, incubator, microscope, centrifuge and cell counter, and distinguish clean air flow from environmental control. Next come primary cultures, finite and continuous cell lines, adherent and suspension systems, confluence, passage and scale-up. We compare dye exclusion, metabolic assays, membrane integrity tests, and cell counting, including a worked viability calculation. Finally, we study controlled rate freezing and revival, authentication, sterility and mycoplasma testing, major applications, limits of extrapolation, consent, animal reduction, genetic privacy and biosafety. The central challenge is selective reconstruction. Inside an organism, cells receive nutrients, signals, mechanical support, waste removal, and interactions with many other cell types. In vitro culture recreates only selected parts of that environment. A useful experiment therefore controls the culture conditions, documents cell origin and passage, measures viability and function, and compares appropriate controls. Normal morphology does not prove identity, a metabolic signal does not necessarily equal cell number, and sterility against visible bacteria does not exclude mycoplasma. Most importantly, an effect in one cell line does not automatically predict a whole patient. Reliable animal cell culture combines biological relevance, technical quality, and disciplined inference. Animal cell culture maintains isolated cells in a controlled artificial environment. The system may preserve useful features of the source tissue, such as receptors, metabolism or production of a specific molecule, but isolation changes context. Neighboring cells, extracellular matrix, circulation, immune signals, and organ-level regulation may be missing or simplified. Culture conditions can also select subpopulations over time. Therefore, every result begins with a clear description of species, tissue, cell type, culture format, passage number, and treatment condition. The model is powerful because variables can be controlled and observed, yet its simplicity is also a limitation. A strong answer states what process the culture represents and what aspect of whole organism biology it cannot establish alone. Animal cell media contain balanced inorganic salts, amino acids, vitamins, glucose or another energy substrate, and a buffering system. Osmolality must remain compatible with cell volume and membrane function. Bicarbonate buffered media are commonly matched to a controlled carbon dioxide atmosphere, while other buffers have different requirements. Serum can provide hormones, attachment proteins, lipids, and protective factors, but its complex and variable composition reduces experimental definition and may introduce contaminants. Serum-free or chemically defined media improve control for suitable cells, but require careful optimization. Supplements, antibiotics, and growth factors do not replace aseptic technique. Medium color can offer a rough pH indication, but reliable culture control depends on calibrated conditions, records, and appropriate measurements rather than visual appearance alone. Temperature influences enzyme activity, membrane behavior, and stress. pH affects proteins, transport, and metabolism, while carbon dioxide interacts with bicarbonate buffering. Humidity reduces evaporation, helping stabilize medium volume and osmolality. Oxygen in a standard incubator may differ substantially from the physiological oxygen experienced by a tissue, so the appropriate level depends on the model. Adherent cells also respond to surface coating, stiffness and geometry. The incubator display is not sufficient evidence that every vessel experienced the intended condition. Temperature, gas supply, water pan, door opening, calibration and vessel position matter. Experimental comparisons should document actual culture conditions and change one defined factor while controlling density, medium, passage, and time. A biological safety cabinet uses filtered directional airflow to protect the work operator and environment when correctly selected and used. It is not an incubator and does not sterilize items placed inside. Work must avoid blocking grills or disrupting airflow. The incubator maintains temperature, gas and humidity but requires calibration, cleaning and contamination monitoring. An inverted microscope allows inspection of cells through culture vessels for morphology, attachment, density and contamination. Centrifugation concentrates cells, but excessive force can damage them. Cell counters and hemocytometers estimate concentration and may be combined with viability staining. Equipment performance, cleaning, preventive maintenance and operator technique are all part of data quality. A sophisticated instrument cannot compensate for a misidentified or contaminated culture. A primary culture is established directly from tissue. The sample is dissected, and cells are released by mechanical disruption, enzymatic dissociation or both, under conditions that preserve viability. Different cell types may survive, attach and proliferate differently, so the resulting culture is often heterogeneous. Primary cells can retain many source tissue characteristics and may provide greater physiological relevance than a long-established line. However, they usually have a finite lifespan, show donor-to-donor variation, and can be difficult to standardize. Tissue source, age, health, collection method, processing time and culture conditions belong in the experimental record. Human material requires appropriate consent and governance. Animal-derived material requires ethical approval and reduction principles. Primary does not automatically mean pure or identical to the intact tissue. After cells are transferred from a primary culture into new vessels, the propagated population is described as a cell line. Finite cell lines undergo only a limited number of divisions before senescence. Continuous cell lines acquire or possess changes that permit indefinite proliferation, often with altered chromosomes, growth regulation or phenotype. Their convenience and reproducibility make them widely useful, but long-term passage can cause drift and laboratory selection. A cell line name is therefore not enough. Record should include source, authentication result, passage number, culture conditions and contamination status. Cross-contamination and misidentification have invalidated many experiments. Short tandem repeat profiling, or another suitable species-specific method may establish identity, while morphology alone cannot. Many animal cells are anchorage dependent and must attach to a treated or coated surface before spreading and dividing. They are monitored for confluence, detached using a controlled mechanical or enzymatic method, counted and reseeded before overgrowth alters behavior. Suspension-adapted cells grow free in liquid as individual cells or aggregates and are passaged by dilution into fresh medium. Scale-up may use larger surfaces, microcarriers or stirred vessels depending on the cell. Suspension mixing improves nutrient and gas exchange, but can introduce shear stress. Both formats have minimum and maximum useful densities. Too few cells may lose survival signals, while excessive density depletes nutrients and changes phenotype. The culture format must suit the biology and the intended product. Passaging prevents cultures from exhausting nutrients or exceeding the density at which physiology changes. Adherent cells are washed when appropriate, detached under controlled conditions, neutralized or collected, counted and reseeded at a defined density. Suspension cells are sampled, counted and diluted. Passage number records transfer events, but different split ratios can produce different numbers of cell divisions. Cumulative population doublings provide a more biological measure of culture age. Experiments should use a defined density range, similar growth phase, and consistent time after seeding. Over-trypsinization, harsh pipetting, prolonged handling outside controlled conditions, and inaccurate counting reduce viability. A passage is successful only when cells recover normal attachment or growth and retain the required identity and function. A dye exclusion assay distinguishes cells with intact membranes from cells that take up the dye, but early damage may not be detected. Metabolic assays measure the conversion of a reagent by cellular enzymes. The signal depends on metabolism as well as cell number. Membrane leakage assays detect released cellular components, while fluorescent probes can examine several states. No single method perfectly defines life, death, and function. A viability percentage requires representative sampling and a clear denominator. If 180 cells exclude die and 20 take it up, the total is 200 and viability is 180 divided by 200 times 100 or 90%. Replicate counts, positive and negative controls, and a second method may be needed. Always name the property measured instead of treating every assay signal as direct cell number. Cryopreservation slows biological change and reduces repeated passage. Healthy cells are collected in a validated freezing medium containing a cryoprotectant that reduces ice damage but can itself be toxic during prolonged exposure. Controlled cooling allows water movement and limits intracellular ice before storage at very low temperature. During revival, rapid thawing reduces time in damaging intermediate temperature ranges, cryoprotectant is removed or diluted appropriately, and cells are placed in recovery conditions. Immediate survival is not enough. Attachment or growth, morphology, identity, sterility, mycoplasma status, and required function should be checked. A master cell bank and derived working bank reduce repeated disturbance and improve traceability. Records link every vial to source, passage, freezing date, and test results. Quality control asks separate questions. Is this the intended cell line? Authentication addresses identity. Is the culture free of unwanted organisms? Sterility and mycoplasma tests address contamination, but one test may not detect every agent. Are the cells behaving appropriately? Morphology, growth rate, viability, and a relevant functional assay address performance. Passage history and genetic stability influence interpretation. Mycoplasma is especially important because it may not produce visible turbidity yet can alter metabolism, gene expression, and experimental results. Routine testing, quarantine of incoming lines, separate handling of unverified material, and documented cleaning reduce risk. Antibiotics can mask contamination and should not become a substitute for asepsis. Only combined identity, purity, and function data support a reliable culture claim. Animal cell cultures support virus propagation and vaccine production, recombinant proteins and monoclonal antibodies, toxicity screening, drug testing and studies of cancer, infection, signaling and genetic disease. Production systems require a characterized cell substrate, controlled medium and process, monitoring of growth and product formation, and tests for identity, potency, purity, and safety. Disease models can reveal cellular mechanisms and compare treatments, but relevance depends on cell origin, genotype, differentiation state, and missing tissue interactions. Three-dimensional cultures and co-cultures can add complexity, yet they still do not automatically reproduce a whole organism. The best application pairs a clearly defined question with a model of suitable biological resolution and confirms important conclusions using complementary evidence. Human-derived cells require informed consent or another lawful ethical basis, clear provenance and respect for permitted use. Genomic data derived from cells can carry privacy implications. Animal-derived cultures should support replacement, reduction, and refinement by using alternatives where possible, minimizing animal numbers, and improving procedures. Biosafety assessment considers the cell source, introduced genes, infectious agents, toxins, aerosols, and disposal. Ethical quality also includes honest interpretation. A treatment that changes viability in one line at one concentration does not establish patient benefit or safety. Results must be limited to the experimental model and supported by further studies. Responsible cell culture joins consent, welfare, biosafety, data governance, reproducibility, and transparent communication. For a 3 or 5 mark CBSE answer, begin with the cell source and culture type. Compare primary cultures with cell lines by origin, heterogeneity, lifespan, and physiological relevance. Compare finite with continuous lines by proliferative capacity and transformation. For adherent versus suspension culture, include attachment, passage, and scale-up. In a viability calculation, show viable count, total count, multiplication by 100, and the meaning of the assay. For cryopreservation, write healthy cells, cryoprotectant, controlled cooling, low temperature storage, rapid thawing and post-revival checks. Distinguish the biosafety cabinet from the incubator. End with authentication, mycoplasma testing, passage history, or the limitation that in vitro results do not automatically predict the whole organism. Close your notes and rebuild the chapter. Animal cell culture recreates selected conditions outside the organism, not the complete body. Media provide salts, nutrients, energy, buffers and growth factors. Temperature, carbon dioxide, oxygen, humidity, osmolality and surfaces shape behavior. Primary cultures retain source features but are heterogeneous and finite. Cell lines arise through subculture. Finite and continuous lines differ in lifespan. Adherent and suspension cells require different passage and scale-up methods. Dye exclusion, metabolic assays, membrane tests and counting measure different aspects of viability. Cryopreservation requires cryoprotectant, controlled cooling, careful thawing and recovery checks. Identity, sterility, mycoplasma status, passage, morphology, growth and function support, culture quality. Applications are powerful, but consent, animal reduction, biosafety, privacy and model limitations govern responsible conclusions.